Cairo, 1881. A group of Egyptian and French researchers carefully unwrap the linen bindings of a mummy that has rested undisturbed for more than 3,000 years. The air in the room is dry and ancient. The silence is the kind that settles into stone over centuries, and then they stop. Because there, beneath the final layer of wrapping, is something none of them expected to find on the preserved body of the most powerful ruler in Egyptian history. The mummy was Ramesses II, Pharaoh of Egypt, commander of the Battle of Kadesh, builder of Abu Simbel, the ruler who shaped an entire civilization and whose name was whispered with reverence across the ancient world for generations after his death. And he carried a gene found in fewer than 2 out of every 100 people alive on Earth today. Later analysis by a French research team confirmed what those researchers saw in 1881. The genetic variant was real. Ramesses II was, by every biological measure, a natural redhead. That single fact raises a question that history has never fully answered. How does the rarest pigmentation gene in the human species end up in the bloodline of the most powerful dynasty in the ancient world? And why is it that when you start looking, you find that same question surfacing again and again, buried in deserts, preserved in frozen burial sites, written in the margins of ancient texts that most people have never read? This is where the story begins. Not in Scotland, not in Ireland, not in any of the places we were taught to associate with red hair. It begins in a Cairo examination room, in a silence that lasted only a few seconds before someone finally said something. But the question that silence contained has been sitting unanswered for well over 100 years. Before we go any further into that silence, I want to say welcome. And if you are new here, this channel is a quiet place for long, careful journeys through the parts of history that tend to get left out of the textbooks. If that sounds like something you want more of, consider subscribing and turning on the bell so these stories find their way to you. And now, let us follow that question back to where it truly begins. There is a gene sitting on chromosome 16 of the human genome that has puzzled evolutionary biologists for decades. Not because it is dangerous, not because it causes disease or dysfunction in any catastrophic way, but because, by every standard rule of natural selection, it should have disappeared from the human population a very long time ago. It is called MC1R, melanocortin 1 receptor, and it is responsible for one of the most visually distinctive traits in our species. Here is how it works. In the vast majority of people on Earth, the MC1R gene functions in a straightforward way. It produces eumelanin, the dark brown or black pigment that colors most human hair and skin. Eumelanin is efficient. It absorbs ultraviolet radiation. It protects the skin. It is, from a purely biological standpoint, a remarkably effective solution to the problem of living on a planet with a sun. But in roughly 1 to 2% of the global population, the MC1R gene carries a mutation. A small change in the genetic code, just a few letters swapped out for different ones. And that small change redirects the entire pigmentation system. Instead of producing eumelanin efficiently, the mutated MC1R gene shifts production toward pheomelanin, the red and yellow pigments. When pheomelanin dominates and eumelanin is largely absent, the result is red hair, pale skin, and a set of biological characteristics that, when you examine them closely, raise a very uncomfortable question about how this gene has managed to survive at all. Because the MC1R mutation does not arrive alone, it brings a package of traits with it. Traits that should, under the logic of natural selection, make survival significantly harder. Redheads burn in direct sunlight faster than any other group of people, by a measurable margin. A person with dark hair might spend 30 to 40 minutes in direct sun before the damage begins. A person with the MC1R mutation might have 10 minutes, perhaps less on a clear day near the equator. Their skin cannot produce enough eumelanin to act as a shield. In a prehistoric world where shelter was uncertain and survival meant spending long hours outdoors, this is not a minor inconvenience. It is a genuine liability. Then there is the pain question, and this is where the biology becomes genuinely strange. The MC1R gene does not only affect pigmentation, it is connected to the body's pain processing system in ways that researchers are still working to fully understand. Multiple peer-reviewed studies have demonstrated that people with MC1R variants experience pain differently from the rest of the population. They are more sensitive to thermal pain, both heat and cold. They require more anesthesia during surgical procedures. A 2004 study by Dr. Edwin Liem at the University of Louisville found that patients with red hair required on average 20% more general anesthesia to achieve the same level of sedation as patients without the MC1R variant. They are also more resistant to local anesthetics, including the kind used in dental procedures, which explains a pattern that many redheads have reported throughout their lives without ever knowing the biological reason behind it. The MC1R receptor is present not just in skin cells, but in neurons throughout the central nervous system. It interacts with melanocortin pathways that regulate pain sensitivity, stress response, and inflammation. This is not a superficial trait. This is a gene that reaches into the architecture of how the nervous system itself functions. So, let us hold all of that together for a moment. A gene that makes its carriers more vulnerable to sun damage. That altered pain processing at a neurological level. That comes packaged with pale skin and a reduced capacity to protect against ultraviolet radiation. A gene that is recessive, meaning both parents must carry a copy for a child to express the trait, which makes its spread through any population slow and statistically unlikely. And yet here it is, carried by an estimated 140 million people alive on Earth right now, persisting across ice ages, migrations, plagues, and centuries of documented persecution. Refusing, quietly, and without explanation. What remains unexplained is precisely this. The standard evolutionary argument for the MC1R mutation is that it helped early humans in northern latitudes produce vitamin D more efficiently in low-light conditions. The pale skin that accompanies the mutation allows more ultraviolet B radiation to penetrate the skin and trigger vitamin D synthesis. In a world of long winters, dense cloud cover, and limited sunlight, that advantage could have been meaningful. But vitamin D deficiency, while serious, typically does not kill a person before reproductive age. It weakens bones. It compromises immune function over time. These are real costs, but they are slow costs. Sunburn and melanoma are faster, more direct threats to survival. The math does not fully add up, and researchers who specialize in population genetics will acknowledge this openly. One theory suggests that sexual selection played a role. That red hair, being visually striking and rare, may have functioned as a signal of genetic distinctiveness that certain populations found attractive. Under this model, the gene spread not because it helped people survive. Bright plumage in birds signals genetic health to potential mates. Whether red hair served a similar function in early human mate selection is a question that remains genuinely open. Another theory, quieter and more contested, suggests that the altered pain response itself may have carried an adaptive advantage we have not yet identified. A different threshold for thermal sensation, a different baseline for the body's stress chemistry. In certain environments, under certain pressures, a nervous system wired slightly differently might have offered something useful that we cannot easily measure from a distance of 50,000 years. And then there is the number that changes everything. The 140 million people alive today who express the red hair phenotype represent only the visible surface of the MC1R story. Because the gene is recessive, the carrier population, people who carry one copy of the mutation but do not express red hair, is estimated to be dramatically larger. Some geneticists working with large-scale genomic databases, including datasets from companies like 23andMe, have suggested that the true carrier population globally may exceed 1 billion people. 1 billion people silently carrying a gene they cannot see, may never think about, and will likely never know is there until a child is born, with hair the color of autumn leaves, and a family suddenly has a conversation about a great-grandparent nobody quite remembers clearly. The gene is not rare in the way we think it is. The flame is not small. It is simply hidden, distributed quietly across the human population, in concentrations that our standard models of genetic inheritance have consistently underestimated, which raises a deeper question. If the gene is this widespread, this persistent, this deeply embedded in the human genome across populations that have been separated by oceans and millennia, then perhaps the story of how it got there is not the simple Northern European adaptation narrative we were taught. Perhaps it is much older than that. Perhaps it begins not in the mist-covered highlands of Scotland or the longhouses of Scandinavia, but somewhere far older, somewhere colder and darker and stranger than any of those places. In a world where modern humans were not the only people walking the earth, where other kinds of people, ancient and underestimated, were already carrying something in their DNA that we would not fully understand for another 50,000 years. That is where we are going next. October 2007. A paper is published in the journal Science that does not make front-page news in most of the world. It does not trend on the platforms that were just beginning to reshape how information moved between people. It is a quiet publication, the kind that circulates among specialists and gets filed into academic databases where most people will never find it. But what it contains is, by any reasonable measure, one of the most profound biological discoveries of the modern era. A team of geneticists led by Carles Lalueza Fox at the Institut de Biologia Evolutiva in Barcelona, working in collaboration with researchers at the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, had been doing something that would have seemed impossible just a decade earlier. They were extracting and sequencing DNA from Neanderthal bones. Ancient bones. Bones recovered from El Cedron cave in northern Spain and from a site in the Monte Lessini region of Italy. Bones that had been resting in the earth, undisturbed, for somewhere between 40,000 and 50,000 years, and buried in that ancient genetic material, in the fragmented and carefully reconstructed code of a species that disappeared from the earth before the last ice age had fully ended, they found something that stopped the room. Neanderthals had a variant of the MCZAR gene, not a damaged version, not a corrupted fragment, a functional variant, the kind that, in a living organism, would have disrupted normal eumelanin production and shifted pigmentation toward the red and yellow end of the spectrum. The kind that, had it been expressed in a living Neanderthal, would have produced pale skin and hair in colours ranging from red to auburn to a deep copper that would have caught the firelight in any cave they inhabited. Some Neanderthals, perhaps many of them, were redheads. And then the detail that changes everything. The variant found in the Neanderthal DNA was not the same mutation that produces red hair in modern humans today. It was different. A separate change in the same gene, the same receptor, the same biological pathway, but a distinct genetic alteration arriving at the same destination through a completely different molecular route. In evolutionary biology, this is called convergent evolution. Two lineages, separated by hundreds of thousands of years of independent development, independently arriving at the same solution to the same problem. It happens with eyes. It happens with wings. It happens with the streamlined body shapes of dolphins and sharks, creatures with no common ancestor anywhere near the point where that shape appeared in both of them. And apparently, it happened with red hair, two different human species, living in overlapping territories during a period when the world was colder and darker and less hospitable than anything most of us can fully imagine. Both developed a version of the same pigmentation shift. Both reduced their capacity to produce dark melanin. Both moved toward paler skin and lighter hair. Both arrived, through different genetic paths, at something that looked, from the outside, remarkably similar. The standard explanation from the scientific community is that this reflects a shared selective pressure, northern latitudes, low ultraviolet radiation, long winters with limited sunlight, and the biological need to synthesize vitamin D from whatever light was available. Under those conditions, pale skin would have offered a genuine survival advantage, and any mutation that produced it would have had reason to persist and spread. This explanation is reasonable. It is grounded in established evolutionary principles. And it may be entirely correct, but it leaves a door open, a door that some researchers have quietly noted without fully walking through. Because Neanderthals and modern humans were not simply neighbours who happened to occupy the same general region of the planet during the same general era. They were something more complicated than that. The genomic evidence, accumulated across more than a decade of ancient DNA research led by scientists including Svante Pääbo at the Max Planck Institute, whose work on Neanderthal genomics earned him the Nobel Prize in Physiology or Medicine in 2022, has established something that was deeply contested not long ago and is now accepted as scientific consensus. Modern humans interbred with Neanderthals, repeatedly, in multiple locations, over a period that may have stretched across tens of thousands of years. The evidence is in us. Between 1 and 4% of the DNA carried by people of non-African ancestry today is Neanderthal in origin. That is not a small number when you consider the scale of what it represents. Billions of people walking the earth right now, carrying fragments of a genome from a species that has been extinct for approximately 40,000 years, carrying Neanderthal variants that influence immune function, metabolism, and yes, in some documented cases, skin and hair pigmentation. One theory suggests that the interbreeding between early modern humans and Neanderthal populations in Europe and Western Asia may have introduced or reinforced MC1R variants into the modern human gene pool. Under this model, some of the red hair we see in human populations today is not the product of an independent mutation that arose in modern humans after they arrived in Europe. It may be, in part, an inheritance, a biological legacy passed across the species boundary during encounters that happened in a world so ancient that no memory of them survives except in the quiet code of our own DNA. This theory is contested. The Neanderthal MC1R variant is, as noted, structurally different from the variants most commonly associated with red hair in modern humans. The direct inheritance model has not been conclusively proven, and responsible researchers are careful to say so. What remains unexplained is why the distribution of MC1R variants in modern human populations does not map cleanly onto the patterns we would expect if the gene had a single, relatively recent origin in Northern Europe. It appears in populations and places that the standard migration models do not easily account for. It persists at frequencies that suggest either a very ancient origin, a very wide dispersal, or some combination of selective pressures that we have not yet fully identified. Archaeologists have noted that Neanderthals were, for most of the period during which they existed, dramatically underestimated. The image that dominated scientific thinking for most of the 20th century, the hunched, grunting, primitive creature who was simply outcompeted and replaced by smarter modern humans, has been systematically dismantled by the evidence accumulated over the past 30 years. Neanderthals buried their dead. There is evidence from sites including Shanidar Cave in Iraqi Kurdistan, excavated by Ralph Selecki of Columbia University in the 1950s and 60s, suggesting that Neanderthal burials sometimes included flowers or plant materials arranged around the body. This interpretation remains debated, but the deliberate nature of Neanderthal burial practices is not. They cared for their dead in ways that reflect something recognizable, something that rhymes. Across the distance of 40,000 years, with what we do, they created art. Pigment use has been documented at Neanderthal sites, predating the arrival of modern humans in Europe. Cave markings in sites including La Pasiega and Ardales in Spain have been dated using uranium-thorium methods to periods when only Neanderthals were present in the region. They used ochre, they used feathers and shells as personal ornaments. They had, in other words, an interior life complex enough to express itself through symbol and decoration. They had brains that were, on average, slightly larger than those of modern humans. They survived in environments of extraordinary harshness for a period spanning hundreds of thousands of years. They were not a failed experiment. They were a deeply successful adaptation to a specific set of conditions, and they left traces of themselves in the genomes of their descendants, which is to say, in us. When you look at a red-headed person today, you are looking at someone who carries a visible, expressed version of a gene that echoes across one of the deepest fault lines in human history. The line between modern humans and the other people who shared this planet with us before we were the only ones left. Whether the red hair gene traveled directly from Neanderthal populations into modern human genomes through interbreeding, or whether it arose independently in both lineages in response to the same brutal environmental pressures of Ice Age Europe. The conclusion is the same in either direction. Red hair is ancient in a way that the word ancient barely contains. It is older than history. Older than the oldest city, the oldest written language, the oldest story anyone has ever set down in a form we can still read. It is a trait that was already present in the world before our species had fully become what we are, before we had fully separated ourselves, genetically and culturally, from the other kinds of people we used to share the earth with. And if it was already burning in Neanderthal hair 50,000 years ago, then the question of how it ended up preserved in an Egyptian royal tomb, frozen in the heart of a Chinese desert, and documented across half a dozen ancient civilizations that had no known contact with each other, becomes something considerably more interesting than a simple story about Celtic genetics. That is the question the desert is waiting to answer. There is a desert in the heart of Central Asia that keeps secrets better than almost anywhere else on Earth. The Taklamakan Desert, in what is now the Xinjiang region of Western China, is one of the largest and most arid landscapes on the planet. Its name, in the Uyghur language, translates roughly as the place you go in and do not come out of. The sand there is deep and dry and ancient. The temperatures swing between extremes that would have made long-term habitation nearly impossible for most of human history. It is, by any reasonable measure, one of the last places on Earth where you would expect to find evidence of a thriving, sophisticated population of people with red hair, light eyes, and facial features that do not belong anywhere near Central Asia. And yet, beginning in the early 1900s, and continuing through systematic excavations that have extended into the 21st century, archaeologists working in the Tarim Basin have recovered something that does not fit inside any conventional framework of ancient history. Hundreds of naturally mummified human bodies, preserved by the extreme dryness of the desert air, buried in the sand for somewhere between 2,000 and 4,000 years, and many of them have red hair, not traces of red. Not ambiguous colouring that could be explained by decomposition or oxidation. Clear, preserved, undeniable, red and auburn and reddish-brown hair on bodies that, by every physical measure, do not belong to any population that conventional history places anywhere near that region during that period. The most famous of these mummies is known as the Beauty of Lulan. She was discovered near the ancient dried lakebed of Lop Nor in the eastern Tarim Basin. She died approximately 3,800 years ago, around 1,800 BCE. She was a woman of middle age at the time of her death, and she was buried with care. Alongside her body, archaeologists found a basket, a comb carved from wood, and grain, specifically wheat, which was not native to the region and had to have been brought from somewhere far away. Her hair was light brown to reddish in colour. Her facial structure, examined by physical anthropologists, showed features consistent with populations from Western Eurasia rather than East Asia. She was buried wearing felt clothing decorated in patterns that would not have looked out of place in Bronze Age Europe. She had been lying in that desert, undisturbed and perfectly preserved, for nearly 4,000 years before anyone found her. Then there is the mummy known as Churchen Man, discovered near the town of Churchen in the southern Tarim Basin. He died around 1000 BCE, 3,000 years ago. He was tall, with reddish-brown hair and a full reddish beard. He was buried wearing an elaborately woven garment, decorated in a tartan pattern, a plaid weave structurally similar to the textile traditions associated with Celtic and Germanic cultures in Europe. Next to him in the burial were the bodies of a woman and a child, all showing similar physical characteristics. The woman had blonde hair. The child was wrapped in a textile of remarkable quality. Archaeologists have noted that the textile technology found in the Tarim Basin burials is, by any standard, sophisticated. The weaving techniques, the dye sources, the structural complexity of the fabrics recovered from these sites reflect a level of craft knowledge that took generations to develop. These were not people passing through. They were communities, settled populations with accumulated knowledge and cultural traditions that they had carried with them from somewhere else a very long time before they arrived in the desert. Victor Mayer, a professor of Chinese language and literature at the University of Pennsylvania, has spent decades studying the Tarim mummies and is one of the foremost Western authorities on their significance. He has argued consistently that these remains represent evidence of early Indo-European populations in Central Asia, populations that arrived in the Tarim Basin significantly earlier than any written historical record documents, and whose presence there has been systematically underappreciated in mainstream histories of the ancient world. The genetic analysis has only deepened the puzzle. A landmark study published in the journal Nature in 2021, led by Chungwon Jong of Seoul National University, and an international team of researchers performed comprehensive genomic sequencing on samples taken from 22 of the Tarim mummies. The results were, by the standards of ancient DNA research, remarkable. The Tarim mummies did not show close genetic relationships with populations from Western Eurasia, as earlier hypotheses had suggested. They did not show significant affinity with populations from the Caucasus, the Pontic steppe, or even with the ancient Iranian or Anatolian populations that have been identified as major contributors to the genetic landscape of Bronze Age Eurasia. Instead, the genomic data suggested that the Tarim Basin population was genetically isolated, ancient, a population that had been separated from other human groups for a very long time before they appeared in the desert. The researchers identified their closest genetic relatives not among Bronze Age Eurasians, but among a much older group, the ancient North Eurasians, a population known primarily from a small number of ancient specimens, including the Malta boy, a child buried in Siberia approximately 24,000 years ago. What this means, in practical terms, is that the people buried in the Tarim Basin may represent a remnant population, a group that split from a common ancestral pool very early in the history of modern human dispersal across Eurasia and then remained largely isolated, carrying their ancient genetic signature forward through time, while the populations around them mixed and changed and transformed into the peoples that history eventually recorded. One theory suggests that they may have been among the earliest practitioners of Bronze Age pastoralism and textile production in Central Asia, possibly contributing technological knowledge to the network of trade routes that would eventually develop into what we call the Silk Road. If that theory holds, then these red-haired people buried in the Taklamakan Desert were not an isolated curiosity. They were participants in the earliest chapters of the long-distance exchange system that connected the ancient world. What remains unexplained is how they got there in the first place. The genetic data rules out the most straightforward migration hypotheses. They did not simply walk east from Europe or south from the steppe in any pattern that matches known population movements. Their ancestry is older and stranger than that. And the MC1R variants present in their remains, those red and auburn and reddish-brown shades preserved across 4 millennia of desert burial, point toward a pigmentation history that predates the migrations we usually use to explain red hair in any population. There is another layer to this story that deserves to be acknowledged directly. For decades after the Tarim mummies began to attract serious scholarly attention in the West, access to the remains and to the genetic data derived from them was restricted. The mummies are housed primarily at the Xinjiang Regional Museum in Urumqi, and for a significant period, international researchers faced substantial obstacles in obtaining samples for independent analysis. The political sensitivity around the Tarim Basin region, which has been the site of ongoing ethnic and cultural tensions between the Chinese government and the Uyghur population, created a context in which the question of who the ancient inhabitants of that region were carried implications that extended well beyond archaeology. One theory suggests that the restriction on research access was in part motivated by political concerns about how evidence of an ancient non-Han population in Xinjiang might complicate contemporary narratives about the region's historical relationship to the Chinese state. This theory is advanced by some Western researchers and journalists, and is contested by Chinese officials and some academic institutions. It cannot be verified from outside the relevant decision-making processes. What can be said with confidence is that the 2021 Nature study represented a significant opening in international access to the genetic material, and that its conclusions, precisely because they identified the Tarim people as an isolated ancient population with no strong ties to any currently living ethnic group, were received as scientifically significant, without generating the political controversy that earlier genetic hypotheses had risked. The beauty of Lulan and the Churchen Man, and the hundreds of other preserved bodies lying in the museums, and the still unexcavated sands of the Tarim Basin, represent a chapter of human history that does not fit cleanly into any existing narrative. They were real people. They had names that no one living knows. They had histories that were never written down in any language we can read. They carried genetic material that connects them to populations so ancient that our entire recorded history fits inside a fraction of the time that separates them from their closest known relatives. And some of them had red hair, in a desert in the heart of Asia, thousands of miles from any place that conventional history associates with that trait. Buried with wheat from somewhere else, wearing textiles from a tradition that belongs somewhere else, carrying a gene that connects them to a story much larger and much older than any of the categories we use to organize our understanding of the ancient world. Archaeologists have noted that the Tarim Basin has not been fully excavated. The sites identified and studied so far represent a fraction of what the desert may still contain. New mummies continue to be discovered. New genetic samples continue to be analyzed. The picture these remains paint is not finished. It is still being assembled, slowly, from bodies that have been waiting in the sand for 4,000 years to be found and understood and given back some small portion of the history they represent. The desert kept its secrets for a very long time. It is only beginning, in the quietest possible way, to let them go. And if red hair could travel this far east, carried by people whose origins we are only now beginning to map, then the question of where else it traveled, and what happened to the people who carried it when they arrived, opens into a territory considerably darker than the desert. Egypt is waiting. And what the pharaohs left behind in their tombs is stranger than anything buried in the sand. The Cairo Museum holds approximately 120,000 artifacts within its walls. Mummies, statues, jewelry, papyrus scrolls, fragments of painted plaster that once covered the interior walls of tombs sealed for millennia. It is one of the densest concentrations of preserved ancient material anywhere on the planet, and most people who walk through its galleries move quickly, pausing at the famous pieces and passing the rest without stopping. But there is a room in that museum where, if you know what you are looking at, something quietly extraordinary is on display. The Royal Mummy Room contains the preserved remains of some of the most powerful rulers in the history of human civilization. And if you look carefully at the hair, which has survived the millennia in a state of preservation that is genuinely difficult to comprehend, a pattern begins to emerge. A pattern that does not fit the image of ancient Egypt that most of us carry in our heads. It begins with Ramesses II. He ruled Egypt for 66 years, from approximately 1279 to 1213 BCE. He commanded armies, built temples on a scale that still staggers the imagination, and conducted diplomacy with the Hittite Empire that produced what may be the earliest surviving peace treaty in recorded history. He was, by any measure, the dominant figure of the ancient world during his lifetime, and his reputation persisted for centuries after his death. Later pharaohs added his name to their own monuments. Ancient writers referenced him as the standard against which other rulers were judged. When his mummy was examined by a French research team in 1976, the scientists who participated in the analysis noted the presence of red hair. The mummy had been transported to Paris for conservation work, and the examination that accompanied that process produced findings that were published but never widely circulated outside specialist literature. The hair was analysed using methods available at the time, and the conclusion was that the colouring was natural, not the result of henna application or post-mortem chemical change. Later forensic analysis confirmed what the earlier examination suggested. Ramesses II carried a pigmentation profile consistent with the MC1R variant. The most powerful ruler in Egyptian history was, genetically speaking, a redhead. But he was not alone. Seti I, the father of Ramesses II and the founder of the 19th dynasty, is described in some historical accounts as having similarly light colouring. His mummy, also housed in Cairo, has hair that forensic examination has characterized as light in pigmentation. The physical similarity between father and son, preserved across more than 3,000 years of mummification, suggests that whatever genetic profile produced the red hair of Ramesses II was inherited, passed down through a royal lineage rather than appearing as an isolated anomaly. Then there is Yuya. Yuya was not a pharaoh. He was a powerful court official during the reign of Amenhotep III, and he holds a particular significance in Egyptian history because he was the maternal great-grandfather of Tutankhamun. His mummy was discovered in the Valley of the Kings in 1905 by archaeologist James Keebel, and it is considered one of the best preserved mummies ever recovered from that site. His hair is described in the excavation records and in subsequent analyses as yellowish-white in the state of preservation, but with structural and pigmentation characteristics that some researchers have interpreted as consistent with originally light or reddish colouring. His facial features were noted by early excavators as distinctly non-Egyptian in appearance, leading to decades of speculation about his origins that has never been fully resolved. Archaeologists have noted that Yuya's name itself is unusual within the Egyptian naming conventions of his period, and that his background remains genuinely uncertain. He appears in the historical record fully formed, a man of enormous influence, whose origins are not clearly documented in any surviving text. What remains unexplained is how a family line carrying physical characteristics this distinct from the broader Egyptian population came to occupy such a central position in the royal dynasty at multiple points across several generations. And then there is the god. Set, known also as Seth, is one of the oldest and most complex figures in the Egyptian pantheon. He is the god of storms, of the desert, of chaos and disruption. He is the figure who, in the most foundational myth of Egyptian religion, murders his brother Osiris and scatters his body across the land. He is the adversary, the force of disorder that must be perpetually controlled and contained by the rightful order of the universe. And in the ancient Egyptian symbolic system, Set was associated with the color red. His hair, in the iconographic tradition, is depicted as red. Animals associated with him, including the Shah, a creature whose exact identification remains debated among zoologists and Egyptologists alike, are shown with red coloring. The word Desha in ancient Egyptian, meaning red, appears in ritual contexts connected to Set, in ways that are not coincidental. Red was the color of the desert, the color of blood, the color of the chaos that existed beyond the ordered boundaries of the Nile Valley civilization. And people with red hair, in certain periods and certain texts, were associated with Set in ways that carry implications far beyond simple aesthetic symbolism. Dr. Joanne Fletcher of the University of York, one of the leading authorities on ancient Egyptian hair and mummification practices, has documented evidence suggesting that in specific ritual contexts, individuals with red hair were selected for sacrifice to Set. The practice, if it existed in the form that some texts suggest, would represent one of the earliest documented instances of a physical genetic trait being used as a criterion for ritual selection, not chosen for their crimes or their status or their relationship to the divine, chosen because of the color of their hair. One theory suggests that the association between red hair and Set reflects an ancient memory of a population that Egyptians encountered and absorbed or conquered and displaced at some point in the deep prehistory of the Nile Valley civilization, a population that was physically distinct, that carried the MC1R variant at high frequencies, and whose difference was gradually mythologized into the symbolic framework of Egyptian religion in the way that historical memory often becomes encoded in myth when the original events recede too far into the past to be recorded. Accurately, this theory cannot be confirmed. The documentary record is too fragmentary and too filtered through centuries of ritual use to support firm conclusions. But the pattern it describes is coherent with what the physical evidence suggests, because the physical evidence presents a genuine puzzle. Egypt is a northeastern African civilization. Its population, across the vast sweep of its history, was predominantly dark-haired and dark-skinned, as you would expect from a culture centered on a river valley in a region with intense solar radiation. The MC1R variant, which produces pale skin and red hair, confers no advantage in that environment. It is, if anything, a liability under the Egyptian sun. The gene should be rare in that population. It should appear occasionally, the product of distant ancestry or genetic drift, but it should not cluster. It should not appear with any statistical regularity in the most genetically selected population in the country, which is to say, the royal family. And yet the mummies of the 19th dynasty tell a different story. One theory suggests that the appearance of MC1R variants in the Egyptian royal line reflects a deliberate or incidental introduction of outside genetic material through the political marriages and diplomatic alliances that were a standard feature of Egyptian foreign policy. The Hittites, the Mitanni, the various populations of the Levant and the Eastern Mediterranean with whom Egypt maintained complex and sometimes intimate political relationships across centuries. Any of these connections could have introduced genetic material from populations where MC1R variants were more common. But this explanation, while plausible, does not account for the persistence of the trait across multiple generations of a royal family that was, by the standards of ancient hereditary monarchy, more likely to marry within a restricted pool of candidates than to continuously introduce outside genetic material. What remains unexplained is why the pattern exists at all. Why this specific gene, in this specific concentration, in this specific dynasty? The answer may be mundane. It may be the product of random founder effects and genetic drift within a small breeding population. It may be exactly what the simplest explanation suggests. Nothing more than chance preserved in remarkable condition by the dry air of the Valley of the Kings. Or it may be a thread that, if followed carefully enough, connects back to something older than the dynasty that carried it. Something that travelled into Egypt, from outside, through pathways that the historical record has not fully preserved, carried by people whose names and origins were absorbed into the Egyptian story so completely that only the gene itself remains as evidence that they were ever there. The Royal Mummy Room in Cairo is open to visitors. The mummies lie in climate-controlled cases, preserved by technology that Ramesses II could not have imagined, lit by lighting designed to protect them from further deterioration. Thousands of people walk past them every day. Most of them do not notice the hair, but it is there. Patient and ancient and silent, still carrying, after more than 3,000 years, the molecular signature of a question that Egyptian civilization encoded into its mythology and its rituals and its royal bloodline without ever, as far as we can tell, fully understanding what it was asking. We are still asking it, and the answer, if it exists, is buried somewhere in the distance between a gene on chromosome 16 and a god with red hair, standing at the edge of the ordered world, looking out into the chaos beyond. Imagine you are a Roman soldier stationed on the northern frontier of the known world. You have been trained in the most disciplined military system in human history. You have marched in formation, maintained your shield wall, learned to move as part of a machine designed to absorb and destroy anything that comes against it. You have been told that the peoples beyond the borders are barbarians, disorganized, primitive. That Roman order will prevail over Celtic chaos, the way it has prevailed over every other force the Empire has encountered on its long march across the ancient world. And then you see them coming across the field, and nothing in your training prepared you for this. The size of them first. Celtic warriors, drawn from the tribes of Gaul and Britannia, and the highland regions beyond Roman administration were, on average, significantly taller than the Mediterranean soldiers who faced them. Roman sources note this repeatedly, and the skeletal evidence recovered from burial sites across the Celtic world confirms it. These were large people, physically imposing in a way that would have registered immediately and viscerally across an open battlefield. Then the sound. A wall of noise designed not to communicate, but to overwhelm. Horns, drums, screaming in a language that contained no words a Roman soldier would recognize, a sound that ancient writers compared to thunder moving across the ground rather than through the sky. And then the hair. Streams of red and copper and deep auburn, some of it natural, some of it enhanced with lime paste that the Celts applied before battle to lighten the colour and stiffen the texture, pulling it upward and outward into shapes that caught the light and moved with the wind in ways that human hair, in any Roman soldier's experience, was not supposed to move. Julius Caesar, writing in his account of the Gallic Wars, described the physical appearance of the peoples he encountered north of the Mediterranean with a specificity that suggests genuine observation rather than rhetorical exaggeration. He noted their great size, their distinctive colouring, and the deliberate way they used their appearance as a component of their military presence. Tacitus, writing about the Germanic and British tribes in his work Germania, observed that red hair was common among the northern peoples, and that it set them apart from the darker-haired Mediterranean populations in ways that Romans consistently noted as remarkable. He was not describing an aesthetic preference. He was documenting what Roman soldiers saw across the front lines of imperial expansion, and what they saw unsettled them in ways that purely tactical analysis could not fully explain. Dio Cassius, the Roman historian writing in the 2nd and 3rd centuries of the Common Era, left what is arguably the most vivid physical description of a Celtic leader in the ancient record. His account of Boudica, the queen of the Iceni tribe who led the most serious rebellion against Roman rule in the history of the British provinces around 60 CE, describes a woman of great height with a fierce gaze, harsh voice, and a mass of bright red hair that fell to her hips. Boudica's rebellion destroyed 3 Roman cities, including Camulodunum, Londinium, and Verulamium, killed an estimated 70,000 Roman citizens and their allies, according to Tacitus, and came closer to ending the Roman project in Britain than any other event in the island's history under imperial rule. She was eventually defeated, but the fact that her physical appearance was preserved in detail by Roman historians, who were otherwise not in the habit of describing the bodies of their enemies in flattering terms, tells you something about the impression she made. Red hair, in the context of Celtic warrior culture, was not a passive biological trait. It was an active component of how these people presented themselves to the world and to their enemies. Archaeologists have noted that the Celtic practice of using lime paste to treat hair before battle is documented in multiple ancient sources and is supported by physical evidence recovered from burial sites across the British Isles and continental Europe. The lime treatment, applied to damp hair and then shaped as it dried, would have produced effects that modern experimental archaeologists have replicated. Hair treated this way becomes dramatically lighter in colour, stiffer in texture, and capable of being formed into shapes that remain stable even during physical exertion. Combined with naturally red or auburn hair, the effect would have been, under any lighting conditions, and especially in the low-angle sunlight of a northern European morning battlefield, genuinely alarming to anyone who had not encountered it before. This was not accident. This was design. The Celts understood, with the kind of practical intelligence that military cultures develop through generations of conflict, that the first seconds of a confrontation are often decisive, that the psychological state of the enemy in those first seconds, before a single blow has been struck, can determine the outcome of everything that follows. They built a visual system of terror around the physical characteristics that their population naturally possessed, and they amplified those characteristics deliberately and methodically. Red hair was a weapon, perhaps the most unusual weapon in the ancient world, and the people who carried it knew exactly what it was worth. But the question of why the Celtic regions, particularly Ireland and Scotland, became and remained the global concentration points for the MC. 1R gene, long after the Roman period ended, requires an answer that goes beyond battlefield psychology. The geography tells most of the story. Europe, across the period from roughly 5000 to 2000 BCE, experienced wave after wave of population movement. Farming cultures spreading from Anatolia, steppe peoples moving westward from the Pontic-Caspian region. Each new wave displaced earlier populations, pushing them toward the margins of the continent. When you reached the western shores of what is now France, you could go no further west on land. When you crossed to Ireland, you had reached the final edge. There was nothing beyond the western coast of Ireland except ocean. This meant that populations carrying specific genetic profiles, including elevated frequencies of the MC1R variant, were effectively compressed into the far western and northwestern corners of Europe. They arrived and they stayed, because there was nowhere further to go. Over generations, the genetic founder effect amplified the traits that were already present in those founding populations. A gene that might have remained at low frequency in a large, mobile, mixing population became common in a small, geographically isolated one. Ireland's position made it a terminal point for genetic flow in a way that few other places on Earth could match. Populations arrived from the east and from the south, and they added their genes to the existing pool, but the island itself retained a core genetic signature that reflected its oldest inhabitants with unusual fidelity. The Highlands of Scotland functioned similarly, with the additional factor of extreme terrain making the isolation of individual communities within the larger region even more pronounced. Small valleys, separated by mountains that were genuinely impassable for significant parts of the year, meant that genetic drift could operate at the level of individual glens and clan territories over centuries. One theory suggests that the unusually high frequencies of MC1R variants in Ireland and Scotland today, with Ireland carrying the highest documented rate of red hair expression in the world at approximately 10% of the population, and Scotland close behind, reflect not a single founding event but a series of successive compressions. Each wave of displacement pushing populations with higher MC1R frequencies further west, each geographic bottleneck concentrating the gene further, until the western edge of Europe became what it remains today, the place where that ancient pigmentation gene is most densely preserved. By the early medieval period, the association between red hair and Celtic identity had become so fixed in the cultural imagination of Europe that it functioned almost as a shorthand. To describe someone as Celtic was, in certain contexts, to imply red or reddish hair. To describe red hair was to invoke the entire complex of associations that Celtic culture had built around it. The fierceness, the independence, the refusal to be absorbed into larger imperial systems without resistance. Those associations would carry forward into the centuries that followed. But the cultural meaning of red hair was about to undergo a transformation that the Celtic warriors, who used it as a psychological weapon, could not have anticipated and would not have recognized. Because the same visual quality that made red hair a source of power on a Celtic battlefield, its immediate visibility, its resistance to being overlooked or ignored, would eventually make it something else entirely in a different cultural context. Not a weapon, not a mark of warrior power, a target. And the people who would turn it into one were already moving across the northern seas, carrying their own version of the flame, spreading it to places that had never seen it before, and setting in motion a chain of events that would reshape the genetic map of an entire continent. The longships are coming. There is a name that appears in the Norse sagas so casually, so matter-of-factly, that most readers pass over it without pausing to consider what it actually tells us. Erik Thorvaldsson. He is known to history by a different name entirely. A name that was not given to him at birth but was attached to him so completely that his original name became almost irrelevant. He is known as Eric the Red, and the reason he is known by that name is not complicated. It is not a metaphor. It is not a reference to his temperament or his reputation for violence, though both of those things were also true. It is a straightforward physical description. Erik Thorvaldsson had red hair, and in the Norse world of the late 10th century, that was considered a sufficiently defining characteristic to become the name by which a man would be remembered across 1,000 years of history. What is remarkable about this is not the red hair itself. What is remarkable is the complete absence of stigma attached to it. In the sagas, Eric the Red is not marked as different. He is not treated as suspicious or supernatural or threatening because of his colouring. He is a man with red hair who also happens to be one of the most consequential explorers in the history of human expansion across the planet. He was exiled from Norway for manslaughter. He was exiled from Iceland for further killings. And then, from the narrow margin of what was left available to him, he sailed west into waters that no Norse navigator had charted, and found a landmass large enough to sustain human settlement. He named it Greenland, established its first permanent Norse colony around 985 CE, and convinced hundreds of people to follow him there. He did all of this as a man whose defining physical characteristic was his red hair, and the sagas record none of it as unusual. The red hair is simply there, part of the description, no more remarkable in the narrative than his height or his temper. This cultural normalization of red hair within Norse society is not a minor detail. It is a significant data point in understanding how the MC1R gene spread across Europe during the Viking Age, in the pattern that genetic analysis is only now beginning to map with precision. A landmark study published in the journal Nature in 2020, led by Eske Willerslev of the University of Copenhagen and an international team of researchers, performed genomic analysis on the remains of more than 440 individuals from Viking Age burial sites across Scandinavia, the British Isles, Iceland, Greenland, and the broader range of Norse expansion. The study represented the most comprehensive ancient DNA analysis of Viking Age populations ever conducted, and its findings reshaped several assumptions about who the Vikings actually were in genetic terms. The results confirmed what historical sources had suggested but never quantified. The Viking Age populations were not genetically homogeneous. They were diverse, reflecting centuries of contact and interbreeding with populations across a wide geographic range even before the expansion period began, and within that diversity, MC1R variants appeared at frequencies significantly higher than their modern global average. Not the majority of individuals sampled, but common enough, distributed widely enough across the burial sites to suggest that red hair was a visible and unremarkable feature of Viking Age communities throughout Scandinavia and beyond. Thor, the most widely worshipped deity in the Norse pantheon, was not described as blonde, The literary and poetic sources that preserve the earliest layers of Norse religious tradition describe Thor with red hair and a red beard. Rautha, the Old Norse word for red, appears in his descriptions with a consistency that suggests this was a fixed attribute rather than a variable one. The most powerful god in the Viking world, the deity associated with strength, protection, thunder, and the defense of humanity against the forces of chaos, was envisioned as a redhead. One theory suggests that this divine association with red hair created a cultural feedback loop within Norse society. If the most powerful and admirable figure in the religious imagination of the culture was red-haired, then red hair within that culture was unlikely to accumulate the negative associations that it was simultaneously developing in other parts of medieval Europe. The Norse were not worshipping red hair as magical or supernatural. They were simply including it in their highest imaginative category of power and worth, and that inclusion may have prevented the kind of cultural prejudice that, in other societies, would have reduced the reproductive success of red-haired individuals and gradually lowered the frequency of the gene in the population. The Norse expansion began in earnest around 800 CE and continued for approximately 3 centuries. Its geographic range was staggering. Norse raiders and traders and settlers reached the coastlines of Western Europe from Ireland to the Iberian Peninsula. They established permanent settlements in Iceland, Greenland, and briefly in North America at the site now known as L'Anse aux Meadows in Newfoundland, Canada, excavated and confirmed by archaeologist Anne Steen Ingstad and her husband Helga Ingstad in the 1960s. They moved east along the river systems of what is now Russia and Ukraine, establishing trading posts and political structures that would eventually develop into the Kievan Rus, the political entity from which modern Russia traces part of its foundational mythology. They served as elite mercenary soldiers in Constantinople, forming the Varangian Guard that protected the Byzantine emperors, and they brought their genes with them everywhere they went. The genetic impact of 3 centuries of Norse expansion on the populations of the British Isles has been documented in detail by researchers, including David Reich at Harvard Medical School, whose work on ancient DNA has transformed the understanding of population history across Eurasia. In areas of heavy Norse settlement, including the Orkney Islands, the Shetland Islands, coastal Yorkshire, and significant portions of Ireland, modern populations carry Norse genetic markers at frequencies that reflect not occasional raiding contact, but sustained settlement and interbreeding across multiple generations. In some of these regions, Norse ancestry accounts for between 30 and 40% of the modern genetic profile, and where Norse ancestry went, MC1R variants followed. Iceland represents the most complete natural experiment in this process. When Norse settlers began arriving in Iceland in the late 9th century, they brought with them a population that included both Scandinavian individuals and Celtic individuals, particularly women from Ireland and Scotland, who came to the island either as willing settlers or as slaves taken during raids on the Western British Isles. This created, within a few generations, a founding population that combined Norse MC1R variants with Celtic MC1R variants in a geographically isolated environment with no subsequent large-scale immigration to dilute the genetic signal. The result is visible today. Iceland has one of the highest rates of red hair expression in the world, comparable to Ireland and Scotland. This is not coincidence. It is the direct and measurable genetic legacy of the specific population mixture that established the island's founding community more than 1100 years ago. The red-haired Icelanders of the 21st century are, in a very literal genetic sense, the descendants of a Norse-Celtic merger that happened on longships crossing the North Atlantic in the 9th century. The Norse expansion into Russia produced a different, but equally traceable pattern. The Swedish Vikings, who moved east, known as the Varangians or the Rus, established themselves along the river routes connecting the Baltic Sea to the Black Sea and the Caspian Sea. Over generations, they intermarried with Slavic populations, and their genetic contribution to the populations of northwestern Russia has been documented in studies examining the distribution of Scandinavian genetic markers in modern Russian populations. The red hair that appears occasionally in Russian populations today, particularly in the northwestern regions around Novgorod and along the historic Varangian trade routes, reflects this ancient Norse contribution to the Slavic genetic pool, Archaeologists have noted that the speed of the Norse genetic expansion during the Viking Age was, by the standards of pre-modern population movement, extraordinary. The recessive nature of the MC1R gene means that under normal circumstances, a rare variant spreads slowly through a population, constrained by the mathematical probability of two carriers meeting and producing offspring who express the trait. The Norse expansion compressed this timeline dramatically. A single red-haired Norse settler establishing a family in a new territory could introduce the MC1R variant into a population that had never carried it. Within 3 generations, carrier frequency in that local population would be measurably higher. Multiply this across thousands of settlements, hundreds of thousands of individual lives, 3 centuries of sustained movement, and the genetic map of an entire continent begins to change. What remains unexplained is the outer boundary of that change. The documented range of Norse expansion is well established by historical and archaeological evidence, but genetic studies examining populations at the edges of that range have occasionally found Norse markers in places that the historical record does not clearly account for. Some researchers working with data from Central Asian populations have identified genetic signals that they tentatively associate with Norse ancestry. Whether these signals reflect actual Norse contact with those populations through trade routes extending further than the documented historical evidence, or whether they reflect older shared ancestry between Scandinavian and Central Asian populations from periods long before the Viking Age, remains an open question. The Norse did not create red hair. The gene was already ancient when the first longship was built. But they did something that no other population movement in European history had done with quite the same speed and geographic reach. They took a gene that had been concentrated in the far western margins of the continent and redistributed it across an area stretching from the North Atlantic to the steppes of Russia. They spread the fire, unknowingly, incidentally, as a byproduct of everything else they were doing, but the flame they carried is still burning in populations across the northern world, a genetic signature written in the hair of people who have no idea they are carrying a legacy that travelled to them on a longship 1,000 years ago. But while the Norse were spreading red hair genes across the northern world with a cultural neutrality that bordered on indifference, something very different was happening in the rest of medieval Europe. Because not every culture that encountered red hair responded the way the Norse did. In other parts of the continent, the same visible gene that Thor wore as a divine attribute was being reframed. Slowly, quietly, over centuries of accumulated fear and superstition, and the particular cruelty that organized institutions can produce when they need someone to blame. The fire was about to become a death sentence. There is a painting. It hangs in the collection of the National Gallery in London, produced in the 13th century, and it depicts a scene that would have been immediately recognizable to every person who saw it in the medieval world: the Last Supper. And one of them, seated slightly apart from the others, his posture carrying a quality of separation that the painter has made unmistakable without resorting to any dramatic gesture, has red hair. It is Judas Iscariot. There is no instruction in any gospel, in any canonical text of the Christian tradition, that describes the hair colour of Judas Iscariot. The earliest Christian art, produced in the centuries immediately following the period it depicts, does not give Judas any consistent physical characteristic that distinguishes him from the other apostles. He is simply one of 12 men, identifiable by his position in the narrative, rather than by any feature of his appearance. And then, somewhere around the 12th and 13th centuries of the Common Era, something changes. Judas begins to acquire red hair. Not in every depiction, not all at once, but with a consistency that builds across the following centuries, until the association becomes so embedded in the visual language of European Christianity that it stops being a choice and becomes a convention. By the 15th century, a painter depicting the betrayal of Christ, who wanted to make Judas immediately identifiable to a largely illiterate audience, had a simple and universally understood tool available: red hair. The audience would know instantly which figure was the traitor. Ruth Mellinkoff, an art historian whose research into the iconography of outsiders in medieval Northern European art was published by the University of California Press, documented this pattern in detail. Her work traced the systematic use of physical characteristics, including red hair, to mark figures in religious and secular art as morally suspect, dangerous, or aligned with forces of evil. The red hair of Judas was not an isolated artistic decision. It was part of a broader visual vocabulary that medieval European culture developed and refined over centuries for the purpose of marking difference as threat. One theory suggests that the decision to give Judas red hair reflected an existing association in medieval European folk culture between red hair and untrustworthiness, and that the religious iconography both drew on and amplified that association. Under this model, the Church did not create the stigma. It inherited a pre-existing cultural anxiety and encoded it into the most widely distributed visual medium of the medieval world, which was religious art, and in encoding it there, it gave the stigma a permanence and an authority it had not previously possessed. What remains unexplained is where that pre-existing cultural anxiety came from. The most plausible answer connects back to the historical memory of the red-haired peoples that Mediterranean and southern European cultures had encountered as enemies and as others across the preceding centuries. The Celts, who terrified Roman legions, the Germanic warriors, whose red-tinged appearance Tacitus recorded as a mark of their barbarian nature, the slaves from northern regions, whose physical distinctiveness made them simultaneously exotic and threatening in Roman markets. These encounters left cultural residues that outlasted the specific memories of the encounters themselves, persisting in folklore and association long after the political circumstances that generated them had transformed beyond recognition. By the time the medieval period was fully established, red hair carried a freight of association that had been accumulating for centuries. Hot temper, moral unreliability, connections to the wild and the ungoverned and the dangerous. These were not conclusions drawn from individual experience. They were inherited frameworks, passed down through cultural transmission in the same way that any stereotype is transmitted, repeated often enough and in authoritative enough contexts that they eventually achieve the status of obvious truth. The Malleus Maleficarum, published in 1487 by Heinrich Kramer, a Dominican inquisitor operating in the German-speaking regions of the Holy Roman Empire, is one of the most consequential documents in the history of European witch persecution. It provided the theological and procedural framework for the identification, prosecution, and execution of individuals accused of witchcraft, and it was referenced by courts across Catholic and Protestant Europe for nearly 2 centuries after its publication. It does not explicitly name red hair as a marker of witchcraft, but the physical characteristics it associates with individuals susceptible to diabolical influence. Pale skin that does not darken normally in sunlight, unusual sensitivity to certain herbs and preparations, physical responses to pain and cold that differ from the norm, correspond, with a precision that is difficult to dismiss as coincidence, to the documented biological profile of individuals carrying the MC1R variant. Archaeologists have noted that trial records from witch prosecutions across Scotland, Germany, France, and the Low Countries during the 15th, 16th, and 17th centuries frequently include physical descriptions of the accused as part of the documentation. Red hair appears in these descriptions with a frequency that exceeds its prevalence in the general population of the regions concerned. It is listed alongside other identifying characteristics: height, age, distinctive marks, occupation, in the matter-of-fact administrative language of judicial record-keeping. It is not always the primary accusation. It is rarely the sole basis for suspicion. But it is there, present in the records, part of the accumulated description of a person that a community had decided to fear. The Scottish witch trials of the 16th and 17th centuries, documented in the records held by the National Records of Scotland in Edinburgh, produced some of the most detailed surviving accounts of the accusations and interrogations associated with this period of persecution. Scotland executed more people for witchcraft per capita than almost any other European country during the peak persecution period, and Scotland, as the population genetics data confirms, carried MC1R variants at some of the highest frequencies in the world. One theory suggests that this correlation is not coincidental, that in a country where red hair was sufficiently common to be visible but sufficiently unusual to mark its carriers as distinct, the cultural machinery of witch accusation found a ready-made visual criterion that required no technical knowledge to apply. Anyone in a Scottish village could see red hair. Anyone could remember, from the folklore and the sermons and the whispered community knowledge that accumulated over generations, That red hair was associated with the uncanny, with the diabolical, with the forces that a good Christian community was supposed to identify and eliminate. The accused did not have to have done anything. They had to look like someone who might. The vampire mythology of Eastern and Central Europe adds another dimension to this pattern. Slavic and Romanian folk traditions dating from the medieval period include consistent descriptions of vampires as individuals with red hair and pale skin. The connection is documented in academic folklore scholarship, including the work of Paul Barber, whose research into European vampire mythology and its relationship to pre-scientific understandings of decomposition and disease was published by Yale University Press. In regions where red hair was rare enough to be genuinely unusual, it became attached to the figure of the reanimated dead, the creature that existed outside the normal boundaries of life and death, that could not be governed by the ordinary rules of the human community. The pattern across these different cultural contexts, the witch trials of Western and Northern Europe, the vampire mythology of Eastern Europe, the iconographic tradition of Judah in religious art, is consistent enough to suggest a shared underlying mechanism. In societies organized around strong in-group identity and high anxiety about deviation from communal norms, a visible genetic trait that marks its carriers as physically different from the majority becomes a surface onto which that anxiety can be projected. The projection does not require conscious organization. It does not require a decision made in any room by any specific group of people who sat down and agreed to persecute redheads. It requires only the ordinary human tendency to find meaning in visible difference, and the ordinary institutional tendency to codify and amplify whatever meanings the culture has already attached to that difference. What remains unexplained is the full human cost of this centuries-long projection. The witch trial records that survive represent only the cases that were formally prosecuted and documented. They do not capture the social exclusion, the economic marginalization, the quiet daily costs paid by individuals whose visible genetic inheritance made them objects of suspicion in communities organized around conformity and fear. Those costs were real. They were distributed across generations of people whose names are not in any record, who lived smaller lives and occupied more cautious positions in their communities because of the color of their hair. The flame that the Celtic warriors had carried as a weapon, that the Norse had worn as a divine attribute, had become, in the space of a few centuries, and through the specific alchemy of medieval European religious culture, something that its carriers had to hide, or survive, or, in too many documented cases, die for. And through all of it, the gene persisted. It did not disappear. It did not fade from the population under the pressure of centuries of stigma. It continued to be carried, expressed, passed down through generations who learned to be careful about what their visible difference might cost them. The flame, as it always had, refused to go out. But even as the church was turning red hair into a mark of suspicion in the villages and the courtrooms of medieval Europe, in the palaces above those villages, something different was happening, because red hair had also found its way into the bloodlines of the people who ruled. And in that context, it was not a liability. It was a crown. There is a portrait in the National Portrait Gallery in London that most people walk past without stopping. It is not the most famous portrait in the collection. It does not have the immediate visual impact of the larger, more theatrical royal paintings that draw crowds and hold them. But if you stop in front of it and look carefully at the figure it depicts, something accumulates in the looking, a quality of presence that the painter has captured with enough precision that it still communicates across 4.5 centuries of distance. The figure is a young woman. She is perhaps 19 or 20 years old in this image. Her skin is pale in the way that the MC1R variant produces, not the fashionable pallor of powder and cosmetic, but the genuine translucent quality of skin that does not accumulate pigment easily. And her hair, rendered in the particular reddish gold that the painter has mixed with care, catches the light in the upper left corner of the canvas in a way that makes it the visual center of the composition, even though it is not the largest element in the frame. It is Elizabeth Tudor, later known to history as Elizabeth I, the Virgin Queen, the monarch who would rule England for 45 years, defeat the Spanish Armada, and transform a relatively minor Northern European kingdom into the foundational chapter of a global empire. and she had red hair, not the elaborate architectural wigs of her later portraits, constructed from the hair of other women and dyed to maintain the image she had built around herself as she aged. The natural hair of the young woman in the early portraits is documented by contemporaries who saw her in person as a deep, warm, reddish auburn inherited from both sides of a family that had been carrying the MC1R variant through its bloodline for at least 2 generations before she was born. Her father, Henry VIII, is described in accounts from his youth as having reddish gold hair that darkened as he aged. The portraits from his early reign, before the famous later images of the broad, heavily jowled king that have become the defining representation of his appearance, show a young man with colouring that sits clearly in the warm reddish range of the MC1R spectrum. He was considered, by the standards of his time, unusually striking in appearance, and the hair was part of that impression. Her mother, Anne Boleyn, is most commonly depicted in surviving portraits with dark hair, and most historical accounts describe her as dark-haired, but several contemporary descriptions mention chestnut or warm brown tones that some researchers have interpreted as consistent with a single copy of the MC1R variant, which can produce subtle warming in hair colour without the full red expression that requires 2 copies of the gene. If Anne Boleyn was a carrier without full expression, then Elizabeth Tudor inherited 1 copy from each parent and expressed the trait fully, exactly as the genetics of a recessive gene would predict. What Elizabeth understood, with the political intelligence that made her one of the most effective rulers in English history, was that her red hair was not simply a physical characteristic, it was a resource. In an era when royal legitimacy was communicated through visual symbolism as much as through legal argument, the appearance of a monarch was a political statement. Every element of how Elizabeth presented herself in her public portraits was calculated. The white-lead makeup that made her skin appear even paler than its natural tone, the elaborate jewels that signalled wealth and power, the clothing that communicated the full authority of the English crown, and the hair. Always the hair. In the later portraits, when her natural hair had thinned and faded with age, she replaced it with wigs of an even more intense reddish gold than nature had originally given her. She chose to be more red in her official image than she was in reality, because the redness had become part of what she was. It signalled the fierce, uncontainable quality that she had built her entire political identity around. It set her apart from the dark-haired Catholic queens of Europe, from the Spanish Habsburgs, who were her political adversaries, from every category of ruler that the conventional imagination of the period could easily place and therefore easily dismiss. One theory suggests that Elizabeth's deliberate cultivation of her red hair as a political symbol retroactively transformed the cultural meaning of that trait in England, at least within the imagination of the ruling class. The same hair colour that was being used to identify witches in Scottish village courts was simultaneously being worn as a crown by the Queen of England. The contradiction was never resolved. It simply existed, as contradictions do, in the space between different social registers that rarely communicated directly with each other. The House of Stuart, which succeeded Elizabeth on the English throne when James VI of Scotland became James I of England in 1603, carried its own version of the MC1R story. Mary Queen of Scots, James's mother, is most commonly imagined in the romantic historical tradition as a pale, dark-haired figure of tragic beauty, but several contemporary accounts and at least one portrait produced during her lifetime suggest hair with distinctly warm reddish tones, not the bright copper of a fully expressed MC1R phenotype, but the subtler auburn that often marks a carrier. James himself is described in accounts from his youth as having light brown to reddish hair with a reddish beard, a description consistent with a moderate MC1R expression inherited from his mother's side. This means that the unified British crown, from the Tudor period through the early Stuart period, was carried by monarchs who bore the MC1R variant across multiple successive generations. A gene present in fewer than 2 out of every 100 people in the general population appeared in the English and Scottish royal succession with a frequency that simple probability cannot account for. Archaeologists have noted, and geneticists studying European royal lineages have subsequently confirmed, that the inbreeding patterns characteristic of hereditary monarchy create conditions that are genetically unusual by the standards of normal human population dynamics. When a small group of families intermarries across generations, maintaining political alliance and dynastic continuity through carefully managed matrimonial connections, the genetic pool from which each new generation is drawn becomes progressively smaller. Recessive variants that might remain hidden in a large, freely mixing population have a significantly higher probability of appearing in expressed form when both parents come from the same restricted genetic background. A study published in the journal Heredity in 2013 by Francisco Ceballos and Gonzalo Alvarez examined the inbreeding coefficients of European royal dynasties across several centuries and found levels of genetic relatedness that, in some cases, exceeded those typically found in first-cousin marriages. The consequences of this level of inbreeding included the well-documented cases of haemophilia in the descendants of Queen Victoria and the physical and developmental abnormalities observed in the later Habsburg rulers of Spain. But it also, less dramatically and less visibly, amplified the frequency of recessive gene expression across a wide range of traits. Red hair has a recessive phenotype requiring 2 copies of the MC1R variant for full expression would have appeared far more frequently in a population where both parents were drawn from a small gene pool with elevated carrier frequencies than it would in a randomly mixing population. One theory suggests that this genetic amplification effect, combined with the founder effect produced by the Norman conquest of England in 1066, which introduced the Norse Celtic MC1R variants of the Norman ruling class into the English royal bloodline at its foundational moment, created a dynastic genetic environment in which red hair expression was substantially more likely than its global rarity would suggest. William the Conqueror himself is not clearly documented as red-haired, but his son William Rufus, who ruled England as William II from 1087 to 1100, carried a nickname that most historians interpret as referring to his reddish complexion and possibly his hair colour. Rufus, the Latin word for red, was attached to him during his lifetime and used consistently in the chronicles of his reign. Richard I, known as the Lionheart, is described in several contemporary accounts as having reddish or light auburn hair, and his physical appearance was considered sufficiently distinctive that it was documented with unusual specificity for the period. The pattern extends east. Ivan IV of Russia, known to history as Ivan the Terrible, is described in accounts from his own lifetime and in portraits produced under his reign as having reddish-brown hair and a notably reddish beard. The Rurikid dynasty from which Ivan descended traced its origins to the Varangian Norse settlers who had established themselves in the river trading posts of early Russia, and the genetic contribution of those Norse founders to the Russian ruling class is consistent with the MC1R variants that the Norse expansion had distributed across the northern world during the Viking Age. What remains unexplained is whether the concentration of red hair in European royal lineages reflects primarily the genetic mechanisms described above, founder effects and inbreeding amplifying a pre-existing variant, or whether there is something additional in the selection dynamics of aristocratic mate choice that favored the trait in ways we cannot fully reconstruct from the available evidence. What can be said with confidence is that the gene which was marking people for suspicion and persecution in the villages of medieval Europe was simultaneously being worn by the people who ruled those villages. The same flame that could get you burned in a Scottish courtroom could get you crowned in a London palace. The distance between those two outcomes was not genetic. It was social. And it was, in the end, as fragile and as arbitrary as every other form of power that human beings have ever constructed to organize the difference between the people who are safe and the people who are not. Below the palaces, the persecution continued. Above the villages, the flame burned on. And in the laboratories of the 21st century, scientists examining the molecular architecture of the MC1R gene were about to discover that the story was stranger and deeper than anyone in either the palaces or the villages had ever imagined. In 2004, a paper was published in the journal Anesthesiology that caused a quiet but significant disruption in the medical community, not because it announced a cure for anything, not because it identified a new disease or proposed a new treatment. It caused disruption because it confirmed, with the rigor of controlled clinical research, something that anesthesiologists had been noticing informally for years without having the framework to explain it. Patients with red hair require more anesthesia. The study was conducted by Dr. Edwin Liem at the University of Louisville, and it was methodologically careful in ways that made its findings difficult to dismiss. Liem and his colleagues recruited volunteers with naturally red hair and volunteers with dark hair, matched for age, weight, and other variables that might influence anesthetic requirements, and measured the concentration of desflurane, a commonly used general anesthetic, required to prevent movement in response to a standard surgical stimulus. The red-haired subjects required, on average, approximately 19% more anesthetic to achieve the same level of sedation as the dark-haired subjects. 19% is not a marginal difference. In the context of surgical anesthesia, where the margin between adequate sedation and awareness during a procedure can be measured in fractions of a percent, 19% represents a clinically significant gap. It means that a standard dosing protocol calibrated for the general population is systematically underdosing a specific subset of patients. It means that red-haired individuals undergoing surgery under standard protocols may have been experiencing greater awareness during procedures than their surgeons realized. Across every operating room, in every hospital, for as long as general anesthesia has been practiced. The mechanism behind this finding connects directly to the MC1R gene and its role in systems that extend far beyond hair pigmentation. MC1R receptors are present not only in the melanocytes of skin and hair follicles, they are distributed throughout the central nervous system, where they interact with the melanocortin signaling pathway, a system that regulates a remarkably wide range of physiological functions. Pain modulation, stress response, inflammation, the release and processing of endorphins, the body's endogenous pain management molecules. The MC1R variant that produces red hair does not merely redirect pigment production, it alters the baseline chemistry of a system that the brain uses to regulate how the body experiences and responds to physical sensation. This is why redheads are more sensitive to thermal pain, both heat and cold. Multiple studies conducted across different research institutions have replicated this finding with sufficient consistency that it is now considered established in the medical literature. The MC1R variant shifts the pain threshold in ways that are measurable and predictable, not dramatic enough to be debilitating in ordinary life, but significant enough to produce consistent differences in how red-haired individuals respond to everything from dental procedures to post-surgical recovery. The dental dimension of this is particularly well documented. Redheads are statistically more resistant to local anesthetics, the kind injected directly into tissue to numb a specific area before a dental procedure or minor surgery. A study published in the Journal of the American Dental Association found that patients with red hair were significantly more likely to report inadequate pain control during dental procedures and significantly more likely to avoid dental care due to anxiety about pain management than patients without the MC1R variant. The resistance to local anesthetics appears to operate through a different mechanism than the requirement for increased general anesthesia, involving the interaction between MC1R variants and the kappa opioid receptor system, rather than the volatile anesthetic pathways studied by Liam. What this means in practical terms is that a red-haired patient sitting in a dental chair, experiencing what they report as inadequate numbing after a standard dose of local anesthetic, is not being dramatic or overly sensitive. They are accurately reporting a genuine biological difference in how the drug is interacting with their nervous system. And for centuries, before the molecular basis of this difference was understood, that accurate reporting was routinely dismissed by practitioners who had no framework to explain it. One theory suggests that the history of red-haired individuals being characterized as difficult, oversensitive, or hysterical in medical contexts, a characterization that appears in clinical literature from as recently as the 20th century, reflects in part the systematic failure of medicine to recognize that its standard protocols were calibrated for a majority population and were quietly failing a minority whose biology was genuinely different in ways that the available tools could not yet detect. The melanocortin pathway, which the MC1R gene influences, is connected to the production and regulation of a molecule called POMC, pro-opiomelanocortin, which is a precursor to multiple hormones and signaling peptides, including ACTH, which regulates the stress response through the adrenal glands, and the endorphins that modulate pain and mood. In individuals with MC1R variants, the altered function of the receptor appears to shift the baseline calibration of this entire system in ways that researchers are still working to fully characterize. Dr. Andrea Bedini and colleagues at the University of Bologna have conducted research examining the relationship between MC1R variants and the body's endogenous opioid system, finding that the altered receptor function produces measurable differences in how the body generates and responds to its own pain management chemistry. This research suggests that the pain sensitivity differences observed in red-haired individuals are not simply a matter of having a lower pain threshold in the way the term is commonly understood. They reflect a different baseline architecture of the body's pain regulation system, one that responds differently to both external stimuli and pharmacological intervention. The cancer connection adds another dimension to the MC1R story that extends beyond pain and anesthesia. The National Cancer Institute has documented that individuals with MC1R variants have a significantly elevated risk of melanoma, the most dangerous form of skin cancer, compared to individuals without the variant. This elevated risk is not simply a function of sun exposure. Research conducted at multiple institutions, including work published by teams at the Massachusetts General Hospital and the Wellcome Sanger Institute in Cambridge, has found that the MC1R variant is associated with increased melanoma risk, even in individuals who limit their sun exposure and even in tumors that develop in areas of the body that receive minimal ultraviolet radiation. What remains unexplained is the precise molecular mechanism connecting MC1R to this sun-independent melanoma risk. One proposed explanation involves the role of pheomelanin, the red and yellow pigment that the MC1R variant produces in abundance, in generating reactive oxygen species within cells. Unlike eumelanin, which acts as a relatively stable photoprotective molecule, pheomelanin can participate in chemical reactions that produce free radicals capable of damaging DNA, even in the absence of ultraviolet radiation. If this mechanism is confirmed by ongoing research, it would mean that the elevated cancer risk associated with MC1R is not solely about sun vulnerability. It is about the chemistry of the pigment itself, generating low-level oxidative stress within the cells that carry it regardless of external conditions. This finding, if it holds up to continued investigation, would represent a significant revision of the standard model for understanding MC1R-related cancer risk, and it would have practical implications for screening and prevention protocols for red-haired individuals that go beyond the standard advice about sun protection. The most contested area of current MC1R research involves potential connections between the gene and neurological function beyond pain processing. Several preliminary studies have suggested that MC1R variants may be associated with differences in cognitive processing, specifically in areas related to attention, sensory integration, and emotional response to novel stimuli. The melanocortin pathway is active in brain regions including the hypothalamus, the limbic system, and portions of the prefrontal cortex involved in executive function and emotional regulation. If MC1R variants alter the baseline function of this pathway in the peripheral nervous system in the ways that the anesthesia and pain research demonstrates, it is biologically plausible that the same variants might produce measurable differences in how the central nervous system processes certain categories of information. What remains unexplained is whether these preliminary neurological findings will survive the rigorous replication process that distinguishes established science from preliminary observation. The existing studies are small, their methodologies vary, and the effect sizes reported are modest enough that they sit at the boundary between signal and noise. Responsible researchers in this area are explicit about the limitations of the current evidence and the need for larger, more carefully controlled studies before any conclusions can be drawn. One theory suggests that if neurological differences associated with MC1R are eventually confirmed by the research, they would represent the most profound implication of the gene story. Not simply a pigmentation variant with some associated physiological quirks, but a biological difference that extends into the architecture of perception and cognition. A different way of processing the world, encoded in a single gene, carried by 1 to 2% of humanity, ancient enough to have been present in Neanderthal populations 50,000 years ago. that is a large implication to rest on preliminary data. The science is not there yet, but the question is open in ways that responsible researchers are no longer willing to dismiss without investigation. What is established, confirmed across multiple independent studies, and accepted as part of the medical standard of care in anesthesiology and pain medicine is that the MC1R gene does more than color hair. It reaches into the body's most fundamental regulatory systems. It alters the baseline chemistry of pain and stress and physiological response in ways that are measurable, clinically significant, and only now beginning to be fully accounted for in medical practice. The gene that ancient cultures worshipped and burned and painted onto the face of a traitor was doing something in the bodies of the people who carried it that none of those cultures had the tools to understand. It was rewiring the nervous system, quietly. For 50,000 years, and the implications of that rewiring are still being written. Sometime in 2007, a story began to circulate through the global media ecosystem with the particular velocity that a certain kind of scientific-sounding claim achieves when it combines genuine novelty with a faint undercurrent of melancholy. Red hair, the story said, was going extinct. By the year 2060, according to various versions of the claim as it spread through newspapers, television segments, and the early architecture of what would become social media, the MC1R gene would have faded from the human population entirely. The last natural redhead would be born somewhere in Scotland or Ireland, and then the flame that had survived ice ages and migrations and centuries of persecution would simply go out, undone not by any of the forces that had tried to extinguish it throughout history, but by the quiet mathematics of a globalizing world in which people were mixing more freely than they ever had before, diluting the recessive gene into invisibility across a population too large and too diverse for it to maintain any foothold. The story was compelling. It had the quality of a good elegy, and it was, in its essential scientific claims, not supported by the available evidence. The origin of the extinction narrative has been traced by several journalists and science communicators to a report attributed to the Oxford Hair Foundation, a research organisation based in Oxford that had been funded in part by the hair care industry. The specific claim, that red hair would disappear from the human population by approximately 2060, was circulated in media coverage around 2007 and attributed to researchers associated with that organisation. When scientists at other institutions examined the genetic modelling behind the claim, they found it did not hold up. The New Scientist, one of the most widely read science publications in the English-speaking world, published a correction and clarification, noting that the extinction timeline had no sound scientific basis. The Oxford Hair Foundation itself subsequently distanced itself from the most extreme versions of the claim, but by that point, the story had completed its journey through the global media cycle and had achieved the status of received knowledge. It appears in casual conversation, in internet forums, in the kind of confidently stated misinformation that circulates without attribution because it has been repeated often enough to feel true. What remains unexplained is why the story spread so successfully and so durably, despite the absence of scientific support. One theory suggests that the extinction narrative served a psychological function for the audiences who received it, providing a framework for experiencing red hair as precious and endangered rather than simply rare, transforming a genetic curiosity into an object of cultural mourning. Whether that psychological function served anyone's interests beyond the general human appetite for beautiful and melancholy stories is a question worth sitting with. The actual genetics of what is happening to the MC1R gene in the global population tells a considerably more complicated and considerably less dramatic story. Red hair is a recessive trait. This is the foundational fact that the extinction narrative consistently fails to account for in any meaningful way. For red hair to disappear from the human population, it would not be sufficient for red-haired individuals to stop having children, or to have fewer children than the population average. The gene would have to be eliminated from the carrier population entirely. Every single individual carrying even one copy of the MC1R variant would have to fail to pass that copy to any descendant. The mathematics of how recessive genes move through populations was worked out by Godfrey Hardy and Wilhelm Weinberg independently in 1908, and the Hardy-Weinberg equilibrium that their work describes has been confirmed by a century of population genetics research. Under normal conditions, without strong selective pressure actively working against a gene, recessive variants do not disappear from populations. They persist in the carrier pool, hidden from expression but present in the genome, waiting for the generational moment when two carriers meet and produce offspring who inherit two copies and express the trait. The carrier population for MC1R variants is, as discussed in the earlier section of this story, vastly larger than the expressed red hair population. If approximately 140 million people worldwide currently express red hair, representing roughly 1 to 2% of the global population, then the carrier population, individuals who carry one copy of an MC1R variant without expressing the full phenotype, may number in excess of 1 billion. Some estimates, drawing on genomic databases including the large-scale sequencing projects conducted by companies like 23andMe and the UK Biobank, place the carrier frequency even higher. A gene carried by 1 billion people does not go extinct on a timeline of 50 years. It does not go extinct on a timeline of 500 years. It persists, carried silently through generations in the genomes of people who have no idea it is there, until the particular demographic conditions that allow its expression recur, and a child is born with red hair in a family that has not seen that trait for 3 or 4 or 10 generations. What globalization actually does to the MC1R gene is not eliminate it. It disperses it, as populations that were previously geographically isolated from each other mix and intermarry in patterns that the modern world makes possible and that the ancient world made impossible. MC1R variants that were once concentrated in specific regions spread into populations where they were previously absent or vanishingly rare. The carrier frequency in those new populations may be low initially, but it is not zero, and it will not remain low indefinitely because the gene, once introduced into a population, persists in the carrier pool across generations until the conditions for its expression are met. One theory suggests that the long-term trajectory of globalization, rather than reducing the expressed frequency of red hair globally, may actually increase the number of places on Earth where red-haired children are born, not by increasing the frequency in any single population, but by expanding the geographic range of the carrier pool to include populations where the MC1R variant was previously absent. A child born in a city in South America or Southeast Asia or Sub-Saharan Africa to parents of mixed ancestry might carry MC1R variants inherited from distant European ancestors through migration chains that extend back several generations, and might pass those variants forward to children who eventually, in the right generational combination, express the trait fully. Archaeologists have noted that this pattern of genetic dispersal through migration and mixing is not new. It is, as this entire story has argued, the mechanism through which the MC1R gene has always travelled. From the ancient populations of Ice Age Europe into the Celtic migrations, from the Celtic world into the Norse genetic pool, from the Norse expansion across the continent and into Iceland and Russia and the Mediterranean, from the European colonial period into populations on every inhabited continent, each movement dispersing the gene further, thinning its concentration in any single place while extending its presence into new territories. What remains unexplained is the precise shape that this dispersal will take over the coming centuries. Population genetics can model probabilities, it can estimate carrier frequencies, and predict the statistical likelihood of expression under different demographic scenarios. What it cannot do is tell you exactly where and when the next generation of red-haired people will be born, or in which cities and which families and which parts of the world that have never historically been associated with this trait. A child will one day appear with copper hair and pale skin, and the slightly different pain threshold that comes with the MC1R variant. The flame is not going out. It is travelling, as it has always travelled, carried by people who do not know they are carrying it, into futures that none of us can see clearly from where we are standing now. The extinction story was wrong in its conclusion, but right in one small way. Something is changing. The map of red hair in the world will not look the same in 200 years as it looks today. The concentrations that now exist in Ireland and Scotland and Iceland will likely thin as those populations mix more freely with the global gene pool. But the gene itself will persist, wandering, patient, ancient beyond any framework we normally use to think about human time, waiting as it has always waited for two carriers to meet. Most people, if asked to draw a map of red hair in the world, would produce something predictable: a concentration over Ireland, a dense cluster across Scotland, lighter frequencies spreading through Scandinavia, perhaps extending into Iceland and the northern fringes of continental Europe. Maybe a faint acknowledgement that Wales and northwestern England carry the trait at elevated rates. And then, beyond those boundaries, essentially nothing. A blank canvas representing the rest of the planet, where red hair is understood to be so rare as to be effectively absent. That map is wrong. Not wrong in what it includes, but wrong in what it leaves out. Because the actual global distribution of MC1R variants, as revealed by decades of anthropological fieldwork, population genetics research, and the large-scale genomic databases that have accumulated over the past 2 decades, is considerably stranger and more geographically dispersed than the standard narrative suggests. Red hair, or more precisely the genetic variants that produce it, appears in populations and places that the conventional history of the trait cannot easily account for. And in some of those places, the frequencies are high enough, and the documentation is clear enough, that the gaps in the standard map become genuinely difficult to explain away. Begin with the group that most people have never heard of. The Udmurt people inhabit a region of the Russian Federation known as the Udmurt Republic, located in the eastern part of European Russia between the Kama and Vyatka rivers, roughly in the territory where the foothills of the Ural Mountains begin their gradual rise from the flat western plains. It is not a place that appears frequently in discussions of world history or global genetics. The Udmurt are a Finno-Ugric people, linguistically related to the Finns and the Estonians, with a cultural and genetic history that is distinct from both the Slavic populations that surround them and the Turkic and Mongolic peoples that inhabit the regions further east. And they have, by the best available anthropological measurements, the highest rate of red hair of any population on Earth. Studies conducted by Russian ethnographers and physical anthropologists, including work documented by M.V. Vitov at the Institute of Ethnography in Moscow, have recorded red hair frequencies in Udmurt populations at approximately 10%, comparable to the frequencies found in Ireland and Scotland, and in some subpopulations potentially exceeding them. This is not a marginal or ambiguous finding. It is a consistent pattern documented across multiple independent observations. What remains unexplained is why. The Udmurt are not Celtic. They have no documented historical connection to the Norse populations whose expansion distributed MC1R variants across northern Europe during the Viking Age. Their linguistic family places them in a completely different branch of the Eurasian human family from the populations with which red hair is conventionally associated. The Finno-Ugric peoples are thought to have originated in a region spanning the Ural Mountains and the western Siberian plain, and their genetic profile reflects ancient connections to populations from that region rather than to the Western European populations where the MC1R narrative usually begins and ends. One theory suggests that the Udmurt red hair represents an independent and very ancient expression of the MC1R variant, one that traces back not to Celtic or Norse ancestry, but to the ancient North Eurasian population that the Tarim Basin genetic study identified as the source of that mystery population's unusual genomic profile. Under this model, MC1R variants were present in the ancient populations of northern Eurasia, long before the migrations that produced the Celtic and Norse peoples of Western Europe, and the Udmurt have preserved a version of that ancient genetic heritage, with unusual fidelity because of their geographic isolation and their historical tendency toward endogamy, marriage, within the community. This theory cannot be confirmed with the evidence currently available, but it is consistent with the genomic data in ways that the alternative explanations are not. Move southwest from the Ural region into the Atlas Mountains of Morocco and Algeria, and the map produces another, unexpected concentration. The Berber peoples of North Africa, known also as the Amazigh, have inhabited the mountain ranges and desert fringes of the Maghreb for thousands of years. They are a diverse collection of communities, speaking related but distinct languages, with cultural traditions that predate the Arab conquest of North Africa in the 7th century CE by millennia, and within certain Berber communities, particularly those inhabiting the more isolated highland regions of the Atlas Range, red hair, blue and green eyes, and light skin appear at frequencies that are strikingly inconsistent with the surrounding populations of North Africa and the broader Mediterranean world. Genetic studies examining the population history of Berber communities have found European genetic markers, including MC1R variants, at frequencies that cannot be explained by contact with European populations during the historical period. The genetic signatures are old. Researchers, including those working within the framework of the Human Genome Diversity Project, have suggested that these markers reflect ancient migrations from Europe into North Africa during the Neolithic period, somewhere between 5,000 and 7,000 years ago, predating any written historical record of contact between the two regions. Archaeologists have noted that the Berber oral traditions themselves contain references to ancestral peoples who came from the sea or from the northern mountains in a time beyond clear memory. Whether these traditions preserve a genuine historical memory of actual migration events, or represent the kind of mythological explanation that all cultures construct to account for their own origins, is impossible to determine. But the genetic evidence for ancient European ancestry in Berber populations is independent of those traditions and does not require them to be literally true in order to be valid. Travel east from the Atlas Mountains, across the full width of the Asian continent, into the mountain regions of what is now Afghanistan and northern Pakistan, and the map produces what may be its most persistently discussed and most carefully contested anomaly. The Nuristani people, inhabiting the remote valleys of the Hindu Kush mountain range in northeastern Afghanistan, include within their population individuals with blue and green eyes, light skin, and occasional red or reddish-brown hair. These physical characteristics have attracted attention from Western travelers, scholars, and anthropologists since at least the 19th century, and they have generated a persistent popular theory connecting the Nuristani to the soldiers of Alexander the Great's army, which passed through the region in 327 BCE during the Macedonian king's campaign into Central Asia. The Alexander theory has a certain romantic appeal. It is also, in its strongest form, almost certainly an oversimplification. Clyde Winters and other researchers examining the genetics of populations in the Hindu Kush region have found that the light pigmentation characteristics observed in some Nuristani communities reflect genetic ancestry that is considerably more ancient than the Hellenistic period, more complex than a single founding event associated with Macedonian soldiers settling in mountain valleys 2,300 years ago. The Nuristani genetic profile appears to reflect ancient Indo-European ancestry consistent with the populations that spread eastward from the Pontic steppe during the Bronze Age migrations that reshaped the genetic landscape of Eurasia between approximately 3000 and 1500 BCE. These were the same broad population movements that carried Indo-European languages across the continent and that deposited the ancestors of the Tarim mummies in the Chinese desert. The light pigmentation characteristics, including MC1R variants, that appear in some Nuristani communities may therefore be a survival of the same ancient genetic package that produced light pigmentation in Bronze Age steppe populations, rather than a legacy of Alexander's army specifically. What remains unexplained is why these characteristics have been maintained at visible frequencies in the Nuristani population over the many centuries since those ancient migrations, while the surrounding populations of South and Central Asia show these traits at much lower rates. Geographic isolation in the deep mountain valleys of the Hindu Kush, combined with the same endogamy patterns that may explain the Udmurt concentration, is the most plausible answer available with the current evidence. The Human Genome Diversity Project, an international research initiative that collected genetic samples from populations around the world specifically to document human genetic diversity before globalization and population mixing erased the signals of ancient demographic history, has found traces of MC1R variants in populations scattered across geographic ranges that conventional red hair narratives do not account for. Isolated communities in the Caucasus Mountains, certain populations in the Levant, small groups in Central Asia, whose genetic profiles reflect the layered complexity of a region that has been a crossroads of human migration for 50,000 years. Archaeologists have noted that every one of these unexpected concentrations tells the same underlying story. Human populations have been moving, mixing, separating, and preserving ancient genetic signatures in ways that our written historical record captures only partially and often not at all. The MC1R variant is old enough and has travelled far enough that its presence in any given population cannot be explained by reference to the familiar narrative of Celtic and Norse migration alone. The standard map of red hair is not wrong. It is simply drawn at too small a scale to show the full picture. When you zoom out far enough to see the Udmurt Republic and the Atlas Mountains and the valleys of the Hindu Kush, alongside Ireland and Scotland and Iceland, the distribution of the MC1R gene begins to look less like a regional European trait and more like what the genetic evidence increasingly suggests it actually is: an ancient human inheritance, distributed across the planet by 50,000 years of movement and mixture and survival, present in populations that have forgotten, if they ever knew, the long chain of human lives through which it travelled to reach them. The flame was never confined to any single place. It was always everywhere, waiting in the most unexpected valleys and the most isolated mountain communities to be found by someone willing to look at the map again with different eyes. And in the laboratories where the final chapter of this story is still being written, the researchers who are looking at that map with genomic tools that would have seemed miraculous even a generation ago are finding that the unexpected places are not exceptions to the red hair story. They are the story. There is a question that surfaces whenever a story like this one reaches a certain point. It arrives in comment sections and in messages from viewers who have followed the evidence across the preceding hours and find themselves sitting with something that feels unresolved. The question takes different forms depending on who is asking it, but its core is always the same. Was any of this hidden? Was the story of red hair, with its ancient origins and its unexpected global distribution and its connections to royal bloodlines and forgotten civilizations, deliberately kept from the public? Is there a version of events in which someone, some institution, or government, or organized body of interests made decisions about what this history would be allowed to mean and who would be permitted to know it? It is a reasonable question to ask, and it deserves a careful answer rather than a dismissive one. Let us begin with what can be established. There are documented cases in this story where access to evidence was restricted in ways that delayed scientific understanding and that had identifiable political or institutional motivations behind them. The most concrete example is the Tarim mummies. As discussed earlier in this story, international researchers faced substantial obstacles in accessing the remains housed in the Xinjiang Regional Museum in Urumqi for a significant period following their initial discovery and early analysis. The restriction was real. Its consequences for scientific understanding were real. Decades of potential research were delayed by barriers that were not scientific in origin. The 2021 Nature study that finally produced comprehensive genomic data from the Tarim mummies was the product of a research team that included Chinese scientists working within a framework that the Chinese government had approved. Its conclusions, which identified the Tarim people as an ancient isolated population with no strong ties to any currently living ethnic group in the region, were diplomatically convenient in ways that earlier hypotheses, which had suggested closer connections to Western Eurasian populations, were not. Whether the specific findings of the study reflect the actual genomic data, or were shaped in any way by the political context in which the research was conducted, is a question that external researchers cannot fully evaluate because they do not have independent access to the raw data. Archaeologists have noted that this situation, in which politically sensitive archaeological material is controlled by a government with interests in how that material is interpreted, is not unique to China or to the Tarim mummies. It is a recurring feature of the relationship between archaeology and political authority across the entire history of the discipline. Governments have always had interests in the past, because the past is used to justify the present. The selective preservation, interpretation, and presentation of archaeological evidence in ways that serve those interests is not a conspiracy. It is an institutional pattern that operates through ordinary bureaucratic and political mechanisms, rather than through secret agreements or deliberate cover-ups. One theory suggests that the most significant form of concealment in the red hair story is not active suppression, but what historians sometimes call erasure by neglect. The systematic failure to record, preserve, or transmit historical information about groups and individuals who did not belong to the categories that the dominant institutions of any given period considered worth documenting. The red-haired slaves of the Roman world left no written record of their own experience. The women accused of witchcraft in the Scottish trials appear in the historical record only through the documentation produced by the institutions that prosecuted them, filtered through the language and the interests of those institutions. The ordinary people of medieval Europe who carried the MC1R variant and lived quietly with the social costs of that visibility, who avoided certain professions and certain communities and certain kinds of visibility because they had learned what visibility could cost, left nothing behind that any archive has preserved. This is not a conspiracy, but it is a form of loss that was produced by systems operating in predictable and identifiable ways. James C. Scott, the political scientist and anthropologist at Yale University, whose work on the relationship between state power and the legibility of populations has influenced a generation of scholars across multiple disciplines, has documented extensively how institutional record-keeping consistently captures what serves the interests of the recording institution and consistently fails to capture what does not. The result, across centuries of this selective documentation, is a historical record that reflects the priorities of power rather than the full complexity of human experience. Red hair, in this framework, was documented when it appeared in contexts that served the interests of the recording institutions. When it marked a powerful ruler, it was noted because rulers were worth documenting. When it appeared in a Roman military account of Celtic enemies, it was recorded because the military encounter was the subject of the record, and the physical description served the rhetorical purposes of the writer. When it appeared in a witch trial, it was recorded because the trial was a formal institutional proceeding that generated documentation as a matter of procedural necessity. What was not recorded is the texture of ordinary life with the MC1R variant across the centuries in which that variant carried social costs that everyone in a given community understood without anyone needing to articulate them explicitly. That silence in the historical record is real, but it is the silence of institutional indifference rather than the silence of active suppression. What remains unexplained is more interesting and more subtle than any conspiracy theory could accommodate. Why does a gene present in the ancient populations of Ice Age Europe, documented in Neanderthal remains from 50,000 years ago, appear in the royal bloodlines of ancient Egypt and in the preserved bodies of Bronze Age populations in the Chinese desert and in the mountain communities of Afghanistan and in the most isolated ethnic group in the Russian Federation, without any single coherent migratory narrative connecting all of these appearances to a common source within the time frame that conventional history works with? The honest answer is that we do not know. The genomic tools available to researchers today are more powerful than anything that existed a generation ago, and they are producing data that consistently complicates rather than simplifies the picture. Each new ancient DNA study adds resolution to the image of human prehistory and simultaneously reveals new details that the existing models cannot fully account for. One theory suggests that the distribution of MC1R variants across the globe reflects a human prehistory that is considerably more interconnected and considerably more ancient than the frameworks built from written historical records can capture, that populations were moving and mixing and separating and mixing again across Eurasia for tens of thousands of years before anyone wrote anything down, carrying genetic variants that we are only now beginning to trace, and that the apparent mysteries in the red hair distribution are not mysteries at all, but simply the visible surface of a deep and complex prehistoric reality that archaeology and genomics are slowly assembling from fragments. This is probably the most accurate available answer. It is not dramatic. It does not require hidden archives or suppressed research or institutional conspiracies. It requires only the recognition that human history is considerably older and considerably more complicated than the portion of it that we have written records for, and that the genetic evidence is showing us the outlines of chapters that we did not know existed because no one from those chapters left us anything to read. The impulse to reach for conspiracy as an explanation for gaps in the historical record is understandable. Gaps feel like they should have causes. Causes feel like they should have agents. And agents, in the framework of conspiracy thinking, are people who made decisions to hide things, which means the gaps could, in principle, be filled if only the hidden material were revealed. But most historical gaps are not the product of decisions. They are the product of time, of the simple and irreversible fact that most of what happens to most people in most places across most of human history leaves no trace that survives long enough to be found. The archive that contains the full story of red hair across 50,000 years of human existence does not exist in any vault or any server or any institution that has chosen to restrict access to it. It exists in the DNA of living people, distributed across 1 billion carrier genomes, written in a molecular language that we are only beginning to learn to read with the precision the story deserves. The concealment, if that word applies at all, was never deliberate. It was simply the nature of time, and time, as the oldest of the red-haired mummies lying in the dry air of their ancient resting places, could tell you if they could speak, keeps its secrets not through any act of will but simply by continuing to pass. The story was always there, waiting in the bones, waiting in the genes, waiting for someone with the right tools and the willingness to sit with the question long enough to begin to hear what the silence was actually saying. We have travelled a long way tonight, from a Cairo examination room in 1881, where researchers unwrapped 3,000 years of linen and found something that none of them had words for, through the frozen valleys of Ice Age Europe, where Neanderthal populations carried their own version of the MC1R gene in hair that caught the firelight of caves that no longer exist in any landscape we can visit, through the Taklamakan Desert, where red-haired people built communities in a place that history forgot to record them in, leaving only their preserved bodies in the dry sand as evidence that they were ever there at all. We followed the gene into the tombs of Egyptian pharaohs and the ritual texts of a civilization that simultaneously worshipped and sacrificed the people who carried it. We watched Celtic warriors turn their biology into a psychological weapon on battlefields, where the colour of their hair was as carefully deployed as any sword. We travelled with Norse longships across the North Atlantic and down the river systems of Russia, watching the MC1R variant spread into populations that had never encountered it before, carried by people who thought they were raiding and trading and settling, but were also, without knowing it, rewriting the genetic map of an entire continent. We sat with the silence of medieval courtrooms where the colour of someone's hair was noted in the margin of a prosecution record. We stood in the portrait galleries of European palaces where that same colour was being worn as a crown. We followed the gene into modern surgical theatres and dental chairs and genetics laboratories, where researchers are still, in the 21st century, discovering what it does to the bodies and the nervous systems of the people who carry it. And now we are here. What does all of it mean? The honest answer is that it means different things depending on where you stand in relation to it. If you carry the MC1R variant and express it fully, if your hair catches the light in the way that this gene produces, then what you have been listening to tonight is, in some sense, your own history. A history that goes back further than any family tree can reach, further than any written record, back to ice and darkness and the first cold winters that made pale skin and altered pigmentation a survival strategy worth preserving across 50,000 years of human time. If you carry the variant without expressing it, if you are one of the estimated billion or more people who hold one copy of this gene silently in their genome without knowing it, then this story belongs to you too, in a quieter way. You are carrying something ancient, something that travelled to you through a chain of human lives so long that you could not count the links if you tried. Something that may not have spoken yet in your hair or your skin, but that is present in your cells, waiting with the particular patience that genes have for the generational moment when two carriers meet and the flame reappears. And if you carry none of it, if your genome holds no trace of the MC1R variant in any form, then you are still part of this story, because the story of red hair is ultimately the story of human diversity, of the endless variation that our species has produced across hundreds of thousands of years of living in different environments and moving through different landscapes and finding different solutions to the problems that survival presents. The MC1R gene is one thread in that story, a particularly visible thread, a thread that has been noticed and documented and feared and worshipped and persecuted and celebrated across the full length of recorded history. But one thread among countless others, each carrying its own ancient freight, each connecting the people alive today to ancestors they will never know, and to a past that is written not in any language, but in the molecular code of the genome itself. Archaeologists have noted, and geneticists have confirmed, and historians have begun to appreciate that the human story is older and stranger and more interconnected than any framework built from written records alone can capture. The red hair story is one small window into that larger truth. One gene, one visible trait, one thread of fire running through the full length of human time, appearing in the most unexpected places, surviving the most determined attempts to extinguish it, carrying forward something ancient and irreducible that does not have a name beyond the name of the gene itself. The flame does not go out. It travels. It hides. It waits in the genomes of people who do not know they are carrying it, through generations of dark-haired and brown-haired and blonde-haired descendants, until the quiet mathematics of recessive inheritance brings two carriers together, and a child is born with hair, the colour of autumn leaves, or copper wire, or the last light of a northern afternoon, and the ancient thing that has been travelling since before history began arrives again in a new face, in a new time, in a world that the people who first carried it could not have imagined. Somewhere tonight, a child with red hair is falling asleep in a room that their parents have made warm and quiet for them. They do not know what they are carrying. They do not know how far it has travelled to reach them. They do not know the Neanderthal caves or the Egyptian tombs or the Viking longships or the medieval courtrooms that form the long history of the thing written into their genome. They are simply a child, falling asleep, carrying a flame that has refused for 50,000 years to go out. And as long as humans keep moving and meeting and passing their genes forward into futures they will never see, that flame will keep travelling, quiet, ancient, patient, burning. If this journey through the hidden history of one extraordinary gene has been a companion for your evening, a gentle presence for the hours between waking and sleep, then you are exactly the kind of person this channel exists for. Feel free to leave a like if the story found its way to you. If you want to share where in the world you are listening from tonight, or if your family carries this flame in any form, and if you would like to support this work and ensure that more of these long, careful journeys find their way into the world, there is a channel membership option here on YouTube that makes that possible. It is entirely optional and deeply appreciated. For now, let the history settle into something quieter. Let the ancient places we visited tonight grow distant and soft. The caves and the desert and the longships and the palaces are all behind you now. Rest. The flame goes on without you having to watch it.